Okay, so today we talk about neural development. Um, and this, I always say this every year, this is easily a whole semester course in neural development. And so we'll just sort of skim some of the surface. And on Friday, on the advanced session, I'll go over how the development of the cortical plate works, the cortical development works in more detail. But for this lecture, what we're going to concentrate on is early development and the establishment of the coordinate system of the brain. The brain actually has a cylindrical coordinate system. If you ever took any physics classes, you know there's Cartesian, you got polar coordinates, spherical polar, and then cylindrical. It's kind of like, oh man, that's really out there, but that's actually the coordinate system of the brain. And so what we want to see from looking at early development is basically how does that coordinate system get established, and what's the reason for sort of paying any attention to that? Well, if you as you learn different parts of the brain, you can sort of place them in different positions on this coordinate system to sort of keep track of where stuff is. It's kind of a mnemonic aid as well. So that's the plan today. And so we'll go over early development, the establishment of the neural plate and the neural tube, and what happens after that. So the first thing in thinking about development that's really important to keep in mind is that that development is essentially created by the group actions of intelligent agents. So individual cells each contain all the genetic material of the organism, every single skin cell, gut cell, brain cell. But more importantly, they actually act like little people that are sort of cooperating to generate the organisms. Completely different than any kind of normal manufacturing process where you sort of control it from the top down. So cells have muscles, so they can actually sort of like contract the top part of them independently from the bottom part. They have actin and myosin inside the cell, so they're active. They can sort of move around. If you look at something like an early pyramidal neuron. It doesn't really look like a pyramidal neuron yet, but it's differentiated enough so that it's decided that it's going to be a pyramidal neuron, and you stick it in a dish. Well, it's pretty unhappy because it's normally in this three-dimensional environment, but it will still behave like a pyramidal neuron. So it'll start off as a little blob, and then it'll start sending out some basal dendrites while it's stuck down to the plate, and it'll send up an apical dendrite, like it'll make a two-dimensional pyramidal cell. So it knows which way is up and down, normally from cues around it. And it will send out an axon. And if you look at that axon, the axon has, if I blow it up, it has a little thing called a growth cone on it. And that growth cone is a little muscular thing that can sort of crawl along. And so if you take an axon, so here's a cell somewhere back here, and put it in a plate, as the growth cone sort of grows along, so these little spikes sticking out of it. And those spikes are kind of on a dish. They'll sort of like go like that, like it's feeling around. If you put an axon in front of it, another axon, it'll actually just lift that axon up over it and grow under it. Now, that's obviously not what it does in real life. In real life, it's in this three-dimensional environment, and what it's doing is it's kind of sniffing its way around, trying to figure out like what to connect up to. So it's got all different little detectors there. If it's trying to go to another structure, it'll sort of feel its way around and say, oh, no, that's not the right thing. I got to go to the geniculate. And so it'll keep going until it finds the geniculate. Then it might make a map in the geniculate by detecting the level of some molecule, so it can sort of tell where it is in the geniculate. You see axons make right turns all the time. They go along, and then everybody says, like, nah, right turn time, and everybody who comes and makes a nice right turn over here. So it's a completely different way of thinking about how to assemble something. You're assembling it from parts, but each part is like a little intelligent agent. So, and just one last little story. This is something that Ramon Cajal saw so many years ago. If you look in the cortex, normally pyramidal cells look like this, but he actually found upside down pyramidal cells. And what happened? Well, the cell would somehow get upside down, but then its friends would tell it, no, you're supposed to put your apical dendrite up that way. And then the axon would come at the bottom and would say, no, you're actually make two right turns and then come out that way. So you can see, that guy got sort of upside down, but he fixed himself. A little intelligent agent fixed himself by communicating with its neighbors. So let's start off with very simple development. We'll start off with, I'm drawing pictures of a frog embryo because it's a little simpler to see what's going on in the frog embryo. If you look at an avian, a bird embryo, humans' embryos are much more like avian embryos, like bird embryos. So birds have a large yolk, and then the developmental movements that I'm going to illustrate take place sort of stuck down to that big yolk. It's basically the same movements, but it's a little harder to sort of see the geometry of the situation. And humans, a human egg has a big yolk as well. So the egg starts off, it's a very large cell, the fertilized egg. It's an extremely large cell. And so the first thing that happens is it basically just starts to divide. So it just divides into more and more cells without actually increasing in size. So it just sort of subdivides. So that's early division. Early division, and it really is just division. It just divides. This is fertilized egg. And then what happens is it eventually forms a sphere, approximately a sphere. And one of the things about development, when you first look at the structures of development, they look so simple. You just wonder, you look at an eyelash or a fingernail, you go like, I wonder how did you get such incredible complexity starting off with something that looks like it was designed by a physicist. But you really start off with a sphere. That thing is called, so what I've drawn there, these are the cells. That's actually a cell. It pretty much looks like a little cube. And I'll show you pictures at the end to show you that I'm not making this up. This is actually what it looks like. So this thing's called the blastula. This is called blastulation. So there's 3 ulas that we're going to talk about. So blastula. So that's the blastula. It's bigger than I drew it compared to the early division, but basically you end up with a sphere. Now what happens is the first of the really big developmental sort of rearrangement movements called gastrulation. And it's worth taking out just a few seconds to describe how this occurs. What's the actual physical mechanism of this? So what happens is an in-pocketing forms in this sphere. So what causes that? So what causes that is, if you look at the cells, here's a cell. It's kind of like a cubey-like thing. And there's a person in that guy, and there's another guy over here. They're all, okay, cell is like a person. Okay, so they're all stuck together. They've got little guy wires that hold them together. But then they also have muscles. So how do you actually sort of make a developmental movement? You basically send a signal to all these guys, like pull your legs in. And so that's what they do. They all simultaneously pull the little muscles that are in the bottom part of the cell, contract those muscles. And they're the same proteins that actually form a muscle, except they're just in the cytoplasm. And so what that will do is everybody will shrink their bottom part of their little cubes, and then what will happen is that will throw the whole sheet of cells up into a dome. And so that's actually what causes, that's the kind of things that cause these movements. And obviously you could go the other way. If everybody contracts their arms, it would make an in-pocketing. So that's the actual physical cause of this movement. But what happens is you get, if we just draw a cross section of it, you get this in-pocketing like this. And that in-pocketing is, the formation of that in-pocketing is called gastrulation. So here's all the cells still. And so now we got 3 layers pretty much. And so these are all the cells again. There they all are. Okay, so that thing is called the gastrula. Gastrula. And so this is called, not surprisingly, gastrulation. Gastrulation. Okay, so it turns out that's defining some of the big parts of the brain. And so this top part here is called the neural plate. And the thing underneath it, right underneath it, that thing is the notochord. So the notochord is what forms the spinal cord. The spinal cord eventually encloses the bone of the spinal cord encloses the neurons in the spinal cord, but it actually starts off initially just below it. So, and then the neural plate, that's actually the cells that will form the whole brain, and the spinal cord. So, so now what happens? This one is less familiar, and so now what happens if we kind of draw like a 3/4 view of the embryo? If I just sort of draw the neural plate as a plate, it isn't, it's obviously it doesn't have sharp edges like that. So what happens now is that the edges of the plate roll up. So that process is called neurulation as opposed to gastrulation. And so what you end up with is when it's partly occurred, you sort of end up with something that looks something like this. Like I said, I'll show you some actual pictures. So here the neural tube has mostly rolled up. So something like that. Now you still have you still have cells here, but the cells here have essentially rolled themselves up into a tube. So that process is called neurulation. So that's the formation of the neural tube, which is our basic cylindrical coordinate system. So here's neurulation. So if we cut a section through this, so this is kind of a pseudo 3D picture. If we cut a section through it here, so just make it go more up, yeah. So if I just sort of slice through it that way, what does that look like? So it's a little strange. So if you look at it, what happens is if here's kind of like a cross section of it, the neural tube rolls up, but it doesn't join up at its edges. There's some stuff left over at the top. It looks something like that. That's a bad neural tube. Let's draw a better neural tube. Okay, so here's the, there's these little ears at the top where it doesn't exactly join up. There's some leftover. And that's called the neural crest. Now, so that's the neural tube. So here's all the cells in the neural tube. And here's the notochord. That's actually so just to map it back here. So, so this thing goes to here, and this neural plate goes to there. That's the correspondence. So the notochord and originally from the neural plate. Now, so what is the neural crest? Well, the neural crest turns out to be very, very important for many things that are involved in the head. So if you look at a worm, what does a worm look like? Well, it's got a whole bunch of segments. And you look at the head, maybe the head has a little bit of stuff sticking out of it, but it's not that different from the rest of the body. The segments all look about the same, and the head maybe has a few little eye spot or something. The vertebrate head is really complicated. It's got teeth, eyes, all these organs, jaw for complicated jaw for eating, throat. There's a whole bunch of complicated stuff going on there. A lot of that comes from the neural crest, like parts of the teeth come from the neural crest. And parts of the bones of the skull come from the neural crest. So that's the neural crest. And so what happens after that, it kind of, if you sort of look at it in an animation, it almost looks like it melts like ice cream down over the edge of the neural tube. And if you look later, a little bit later in development, what you see is you've got the neural tube here completely closed, and then you can see these collections of cells off to the side, and those are called the dorsal root ganglia. So there's a bunch of cells in here and a bunch of cells in here, and those things are the actual DRG. You'll see that a lot, dorsal root ganglion. And those are cells that basically eventually will develop into the receptors on your skin. So they actually, what they'll do is one end will go out to the skin. This is the skin here. This thing will turn into the skin eventually. And so one end will go out to the skin, form a receptor. The other end of the cell will send an axon into the nervous system to connect with the nervous system. So that's the dorsal root ganglia. In the spinal cord, that's what the neural crest does. But when you go up into the head, the neural crest does a whole bunch of stuff. Quite complicated. So now you've got underneath that, you still have the notochord here, which it hasn't differentiated much yet, and here you've got segments of the body, the lungs and the heart, other things like that. Okay, so that's a little bit later. So that's dorsal root ganglion that came from the neural crest. So one of the things about cells can count. They're like little people, and they can count up to something like 16. They literally can count exactly up to 16. And they better be able to count when you think about it, because if you actually didn't count correctly, something would be twice as big, because every time you divide, if all the cells are dividing, and they do an extra division, you got twice as many cells. So to accurately control the size of your finger, you have to be pretty good at counting in order to accurately control the size of the brain. Now, that also implies that it's actually not that hard to make the brain bigger, as long as you have some metabolic support for it, because all you do is just tell the guys to count up one more time, count up to 16 instead of 15, and you got that part of the brain is twice as big. So cells can count. It's absolutely critical that they count, and you can actually sort of see, if we look at a slightly, later point. So here, that's it's supposed to be red. So that was the original formation of the neural tube. That's what it looks like, and then a little bit later in time, after the neural crest has kind of gone over and the neural tube has sealed up. And this is the ventricle here. So that's going to be the ventricle in the brain, same ventricle. So what about a little later? So if you go a little later, what you see is you get something that kind of looks something like this. So here's the ventricle. So now the ventricle is kind of small. It's quite small. This is the ventricle right here. It's an opening. And then what you see is that there are all these radial glia, and so they're like little guy wires that go up and attach to the top of the brain. So these are radial glia. And the way the counting is done is that the cells, the things that are going to turn into neurons, they don't look like much. They're just like little blobs. They don't have dendrites or axons or synapses or anything like that. They just look like little blobs. And what they do is they look like little things like this, little blobs like that. And they just crawl up and down these guy wires. And so they crawl up to the top of the guy wire, and then they turn around, and then they crawl down the same guy wire or a nearby guy wire, and when they get to the ventricle, they divide. So now there'll be a cell division, and so now there's two of them. Then what happens is one of them is now what we call born, and so what does that mean? Well, this guy's not going to divide anymore ever, and so this guy will sort of crawl his way out and start piling up down here. So there'll be like a pile of cells down here. So, this whole thing will be filled down here. And these guys are called post-mitotic cells. Post-mitotic mean they're no longer cell dividing. So like I said, this guy, meanwhile, the other one, will continue dividing, and it will crawl back up again, and then crawl back down, divide. One then is born and no longer divides. So that's so they got like just counters in them, little people counters. So if you remember, like 7 plus or minus 2, you can only remember 7 things. Well, cells can remember 16 things, so they can actually accurately count up to 16. So this, and how is this studied? So one way that you can study this is if you put a radioactive tracer into an embryo. What happens is it will get taken up, say something that sort of forms RNA or DNA. It'll get taken up by the cells, and the ones that were just born, since they're not dividing anymore, won't dilute it. And so you can actually sort of stage all the different places where cells have been born. and just by looking for the places that have a lot of radioactive tracer, because the ones that are continuing to divide, they just divided it half and half and half and half again, and so you get much less. So if you just look for the strongly labeled cells, those are the ones that were born, and so you can. That's how you actually study where the cells go to and where they come from in the nervous system. So that's early development. So at this point, usually what I do is I talk a little bit about the development of the eye. So, and the development of the eye turns out to actually come from the brain. So the actual retina it's actually a piece of the brain. That's not true with something like the cochlea. Like the cochlea is actually comes from something that comes from what's something called a placode, which is something that forms from the skin-like tissue. But the retina itself is actually something that develops directly from the brain, an outpocketing of the brain. And again, when you look at these pictures, you think I'm kind of making it up how simple it is. But if you actually look a little bit later, so just to sort of put this into context, so this is the neural tube. It's heading off sort of in this direction. Yeah, so that's a long cut through the neural. It's a perpendicular cut through the cylindrical coordinate system of the neural tube. So how does the eye develop? So the eye, if we look at a little bit later point in time, you have something called the eye cup, and it just sort of what happens is a little bulge comes out. So here's the cells, like that. So this is the eye cup. So that's the neural tube. So basically a little outpocketing comes out, and then the outpocketing gets a little dimple in it. Again, using all these, what's causing that is all these cells just all gathering together and pulling in their feet or pulling in their arms, little people in there. So this is called the optic cup. Sorry, I didn't make that topology correct. There we go. And so this is going to form the retina and the pigment epithelium. So if we actually look at what's what. So basically, so this thing here is the retina, and this thing here is the pigment epithelium. The pigment epithelium is this thing. We'll talk about it again when we get to the retina. It's something that sort of nourishes the retina. If you get a detached retina, that means that your retina has come loose from the pigment epithelium. Bad thing, because the retina is very metabolically active. It needs to be supplied by the pigment epithelium. In humans, the pigment epithelium is like black felt. It absorbs light. In a cat, it's reflective. That's why sort of cats have that eye shine. So I always talk about this to just point out how difficult it is to really figure out how development works. Let's put the skin back over. There's some skin over here, so this is happening under the skin. So it's really hard to figure out how development works. And one example is just looking at a Siamese cat. So if you look at, so what is a Siamese cat? So just think of a Siamese cat. So you got a Siamese cat. It's got, it's not a very good Siamese cat. There. So what is a Siamese cat? Well, it's sort of dark. It's got a dark tail, and it's got dark tips of its ears. And it's got dark little footies. It's got a dark nose. Those are all parts of the body that are actually cooler. And the body of the cat, and like the actual sort of muscles of the legs, those are sort of light tan. So what's going on there? Well, they figured out that basically what's going on is there's a defect somewhere in the melanin pathway. And it turns out you can get a Siamese cat, and it's a temperature-sensitive mutant. So what does that mean? It just means that the gene makes a protein that doesn't work when you warm it up. It unfolds because the protein is not stable. But it turns out it can be just about anything. So here's one of the, I won't actually label that, but here's different intermediate chemicals. And there's an enzyme that causes each of these reactions to take place. So that this is, an enzyme, a gene coding for that enzyme. It turns out you can break any of these enzymes, like 5 or 6 of them, and if you break them, you'll get a Siamese cat. Among other things, its eyes are messed up. So Siamese cats are kind of cross-eyed. And why is this? It's because the axons coming out of their eye made a wrong turn. And so how could that possibly be related to melanin? So it turns out the way this works is this little stalk right here dies. And that's another thing that happens in development a lot. It's called apoptosis. Apoptosis is programmed cell death. And so cells will sort of kill themselves for the betterment of the whole organism in a programmed way. And so that stalk dies, and then what happens is the retinal cells grow down through that debris, and then they cross over and they synapse on the opposite side of the brain. And that's what's messed up in a Siamese cat. The crossing doesn't occur properly. It's supposed to, in a cat, some of them are supposed to cross, and a few of them are supposed to stay on the same side, and the ones that are supposed to stay on the same side don't, and they go and they cross anyway. So melanin causes that. So something about the melanin in dying cells in this stalk here is used to direct some of these axons to stay on the same side of the brain. So that's a classic example of you've got like a melanin gene down here. What does it do? So a lot of times you'll see in the literature, now we found a gene for something. Well, here's a gene. It's transitioning this chemical to this chemical, which is part of the melanin pathway. You blow it out, or sort of like mutate it so it doesn't work as well. It doesn't, that's why the melanin is not being produced in the warmer part of the cat's body. That's why it has that sort of buffy color to it. You break it. It obviously breaks melanin. So there's melanin is used for a lot of different things, but then it messes up the partial crossing of your eyes. So there's a completely wackily different, functions. They're both involving melanin, but that's the kind of thing that makes development hard to figure out because it's so heavily optimized. You don't have that many genes. You got about 20,000 genes, and you got to make everything in your brain and your stomach and your esophagus, and there's all that with just 20,000 genes. And flies already have 14,000 genes, and bacteria already have 8,000 genes. So you only have about twice as, or 6 or 8,000. So you only got 2 or 3 times as many genes as a bacterium, and you got this whole brain and eyes and everything like that. So it's really highly optimized. So always be very wary when somebody is mapping a given gene down to some individual function. So they're probably wrong. Okay, so let's talk a tiny bit now about some other. Oh, let's finish the eyes first, and then I'll talk. So what happens a little bit later in this picture here? So what happens a little bit later is we have so a little bit later you have a situation where the optic cup has sort of budded off. So this is the retina and the pigment epithelium, and here's the neural tube. And so here's all the cells in the pigment epithelium in the retina. And so then what happens is that little optic cup somehow tells the skin to form a little impocketing. It's called a placode. It's actually the lens placode. So there's little impocketing forms like that. And then if you look yet later, what happens is, so here's this a little bigger. You get a nice sort of semi-spherical lens with a bunch of cells in it that came from this thing. So that's actually the lens. And then the skin just grows over the top again, like that. So here's all the skin cells. That's skin. So this guy, so there's skin, retina here, pigment epithelium here, lens here. But you might be looking at this and thinking like you can't see out. So here's the skin covering over. So how do you get to see out? Well, what happens is all the cells right along where your eyelids are commit suicide. So these guys just kill themselves in a little line, like right across the skin going over your eye, and that forms your eyelids. And a very similar thing happens to form your fingers. You get a little paddle of cartilage cells, and then what happens is the cells in between your fingers kill themselves, and then you separate your fingers. That's how you sort of differentiate your fingers. So this is apoptosis occurring here to form your eyelid. So this is your eyelid. It's kind of amazing. So that's formation of the eye. But let's just talk a tiny bit about the fact that, again, this is just emphasizing the fact how development is so optimized and difficult to really fish apart. So if you look at eyes, for a long time it was thought that there were two separate kinds of eyes. So there's rhabdomeric eyes, and what they look like, if you look at the photoreceptors, they look like something like this, and the membrane is thrown into a whole bunch of folds to increase the surface area of the membrane. And there's a big one in the middle, and there's the nucleus. And so the photopigment, the photopigment is here, and these are the kinds of eyes that you have in an insect, like with Ommatidia, where they have like a whole bunch of little eyes, like a bee eye or something, where you see like a sphere, and there's a whole bunch of little segments in it, and each one is a little mini eye, and it has these kinds of photoreceptors in it. And it contains something called R-opsin. And opsin is is the thing that holds the photopigment. So it's a protein that the light-absorbing pigment can sort of stick to, and that it's involved in actually causing an ion flow in order to detect the light. So that's a rhabdomeric eye, but then you have a ciliary photoreceptor. So these are photoreceptor type. And the ciliary photoreceptors do it the other way. So what they do is they make all the folds sort of perpendicular to the light, like that. And that's what we have. And why are they called ciliary? Because they've actually got the remnants of a cilia apparatus in them. So, that's something that sticks out of the cell in waves. It's kind of the stuff that gets, if you breathe in a bunch of dust, how does it get out? Well, the cilia just slowly work it out. It might take a couple weeks, but, eventually you'll cough it up if you're not wearing a mask, and you can get it out. So that thing is still there in a photoreceptor, the actual sort of ciliary body. And these contain a different protein, ciliary opsin. But turns out, I don't know, about 10, 15 years ago, they went and looked at this little, they found a little polychaete worm. And it didn't have a very good eye, or eyes, but it turned out this thing had both kinds of opsin. R opsin, and it also had the C opsin. And so then people realized, both of these things, way back when, were already both there in a single organism. So it looked like, well, maybe these things weren't, quite so different. And then what was found out, so basically, the original idea that, vertebrates had this, and insects had this thing, insects, etc. But then people said, well, why don't we look in humans? And so it turns out the insect opsin is there in humans, still there. And so where is it? So if here's the photoreceptor, so there's the photoreceptor with all those things, and there's something called a bipolar cell that sort of connects up, and then that connects up to a ganglion cell. So here's a ganglion cell. Those are the ones that actually sort of I'm trying to draw the dendrites there. They actually project into the nervous system. So that's a ganglion cell. So not all ganglion cells have it, but it turns out there's actually R opsin in the ganglion cells. So we've got sort of these mosaic insect vertebrate eyes, and it turns out to be quite important. It actually projects up into the nervous system and eventually synapses on the blobs. And the blobs are some structures in V1 that are particularly involved in calculating the brightness of objects. And so there's an example of just the way the things, it's complete. So you start off with a terrible eye. The polychaete can barely see. It's just got a couple little eye spots, not very many neurons in there. But they already had both types of opsins. And then, vertebrates heavily developed this C opsin, but they didn't throw this one away. Let's keep it. And so basically, these are light-sensitive ganglion cells. And normally, these guys, the photoreceptors, pick up the light. But it turns out that some cells are actually photoreceptive in the ganglion cell layer. Okay, so just one last thing. Let's just talk a tiny bit about segmentation. So one of the things that you can see in the vertebrate system is you have segments. So you have, for instance, vertebrae. Those are segments, and those are actually pretty similar to what the segments are in something like a fly. And so a number of years ago in flies, people discovered these things called homeotic genes. And these are genes that one of the first ones was called bithorax. So that doesn't sound good. How do you do this? You sort of irradiate some flies, reproduce them, and you look, oh, there's a really weird one. It has two pairs of wings. Flies normally have just one pair of wings, and they have one pair of really small things called halteres, but bithorax had two pairs of wings like a dragonfly. And then there was another one called antennapedia. And that doesn't sound good. Antennapedia. And so what happened there is you'd have a fly head, there's the tongue of the fly, eyes of the fly, and then where the antennae are supposed to come out, instead you've got a leg. So it was very clear that these genes are somehow involved in the specification of what each segment is supposed to do. And so if those genes get messed up, they actually cause the creation of all the downstream genes that will make a leg to just go and do their own thing, except they're stuck to the head, stuck to the wrong segment. The head is a segment. Instead of they should be, the antenna should be there, not a leg. The leg should be stuck to one of the thorax segments. So it turns, and so those genes in vertebrates, so those genes are in vertebrates, and those genes are called Hox genes in vertebrates, and the same genes. There's even more of them. There's like twice or three times as many of them in vertebrates. And what they do is they specify, if this is the spinal cord, they specify all these segments called somites. And so there's a series of segments, and the most anterior somite forms the eye muscles, but the ones back here, form like if you eat a fish, you sort of see the segments of the muscular segments of each. If you sort of pick apart a fish like that, you see those little segments like that. Each one of those is a somite. That's controlled by the Hox genes. And what are the Hox genes? They're just genes that make a protein that sticks to DNA. And so it sticks to other part of the DNA in order to specify, how to make whatever you need to make back here and how to make an eye muscle up here. But there's another set of segments, and they're related to the jaw. And these originally came from gill arches. And so, 1 minute. Okay, so these are 4 minutes. Okay, so these guys are the gill arches, and these are controlled. So the Hox genes basically control the somites, and then there's a whole nother set of homeotic-like genes called DLX genes, and these control the gill arches. So the first gill arch is the jaw, and then some of these other gill arches form stuff down in the throat and the larynx. So the larynx is back here. So it's important for speech. So that comes from these another set of sort of high-level genes that control other genes. So that's pretty much how the segmentation of the nervous system gets set up. So let's go over quickly to look at a few pictures of what this looks like. So David will go over to change over. So I have to press the overlay only. Okay, so what we're looking at here is Okay. Here you can see there's a little depression here. That's when the neural plate just starts to roll up. And here's the neural plate. So it's just like I said it was. So that thing is the neural plate. And you can see it's starting to come this way like that. And if we look down a little further, erase. So if you look down a little further, you can see it's not quite closed there. And then you can see the notochord down there. And you can see the skin. This is the actual layer of the skin sort of going over the top. So if we look even a little later, we can see how get that guy out of there. Okay, we can see now, this is that thing I drew where the neural tube has closed itself back up again, and there's the notochord. That's the notochord, and then these are somites off to the side. And there's skin going over the top. So that gives you some idea. You know, it really kind of does look like this extremely generalized picture. And so if we look a little later, that's a little later picture of the entire embryo, and you can see where the neural tube is completely closed here, and then you can see into the ventricle over here, and then this over here is a later stage where it's completely closed, and those are the somites. And in the last 10 seconds, let's look at a slightly later picture. And so then again, you can see, you can see the neural tube and somites, and the notochord down here. So the somite itself forms a little tube of its own. So it actually does look like this. And we'll in later lectures we'll look at some pictures when it looks slightly more like a brain. This doesn't look much like a brain. Okay, just under the wire.